All right, so in the interest of time, I think we'll get started. Dr. Ravi, if you can just confirm if my voice is clear.
Yes, Deepak, it is clear.
All right, thank you. So very warm welcome to all the participants, attendees, panelists, and speakers. Welcome to APA India 2021 webinar series. This is our webinar number 14. And the monthly theme is biotherapeutics development and the discovery and biotransformation session. So I think all of you have been with us for almost since last, I would say, little close to 4.5 months. This has been a very long webinar series of 18 webinars in a row on every second, third, and fourth Saturdays, starting from June. So I would like to, first of all, thank all the participants to be with us for such a long period. And it has been a real learning journey for all of us. So thank you. And we will continue this until webinar number 18, which ends in November last Saturday, which is 27th of November. So very quickly, as we start, a special thanks to all of our organizers, the presiding chair and chair-elect of all the committees for their untiring efforts and commitment and involvement, active participation, because of which the very interesting agenda we could put up on board. So big thank you to all the presiding chair and chair-elect of different committees. The chair-elect and the presiding chairs, they are supported by a very active organizing committee. So big thank you to all the committee members in regulated bioanalysis, discovery bioanalysis, biotransformation, chemistry, manufacturing, and CMC. So a big thank you to all of our organizers. We would also like to extend our thanks and gratitude to our sponsors, Shimatsu, SciEx, ZevSci, Phenomenix, Thermo Fisher, Agilent, Syngene, USP, NATCO, Vida Clinical Research, Bristol-Myers Squibb, Zoetis, Nanotemper Technologies, IVL, In Vitro Med Labs, Origin Discoveries Technology Limited, Vuxi Aptech, and Loris Infosystems. So a big thank you to all of our sponsors for their support, and just for all of participants who are joining for the first time today. This is like our 12th meeting in a row. So 12th annual meeting. So all the support, all these sponsors have been supporting AP India chapter since almost a decade. So would like to especially thank all of our sponsors for their continued support. This year, we have collaborated with the Life Sciences Sector Skill Development Council, which comes under Ministry of Skill Development and Entrepreneurship, Government of India. So big thank you to our collaborator as well. Just a couple of announcements are there before we start the session. So for the poster session, we received close to around 45 poster abstracts. From that, we shortlisted around 20 poster abstracts, and all of them have already received the confirmation from the admin team. And we would be actually doing a final poster session on 13th of November. And before that, there'll be just a mock event, mock trial event. So that invite all the poster presenter can expect within next week. So each one of you will be having an 8-minute slot, 6 minutes for Q&A, and 2 minutes for Q&A. May I request everyone to keep your lines muted, please? Thank you. So in total, there will be 9 poster winners out of those 20. So 3 best poster winner prize for first, second, and third. So like that, there are 9 interesting, there are cash rewards, there'll be some trophy, certificate, and a few other surprising prizes. So would like to wish all the participants a good luck in advance. We also, at the end of each webinar, we do this lucky draw kind of thing. So congratulations to all the winners. 13 webinars we have done. So around 26 winners we already listed out here. So would like to congratulate all those winners as well. And would request everyone to please be there till the end of the webinar so you can participate in the draw today also. And for all the winners, would like to just reassure them that within this week, the next week, they will be receiving finally their surprise gift from the Amazon, because there were some uh, administration issues with the Amazon. So now it is being sorted out. So those Amazon gift card will be actually in your mailbox by next week, of course, by Tuesday itself. So after Tuesday, if you did not receive any such email in your email box, please revert back to bostondb21@gmail.com, and we should be able to help you out. So that's about the prizes. Then very quickly, all the attendees, your video and audios would be muted. Are not audible. So if you have any questions for any of the speaker, feel free to use the Q&A tab at the bottom of the screen. During the webinar, if you're not able to clarify your doubts or queries, do write to us at bostondb21@gmail.com. And as I mentioned, please attend all the 3 talks. And towards the end of after panel discussion, we'll be doing a Kindle draw. Finally, all the webinar talks, all the 14 webinars, in fact, all the 18 webinars which is going to take place, all the talks would be uploaded on Boston YouTube channel. So later on, with due consent from each of the speakers. So do subscribe Boston International channel if you want to refer any of the talks later on. So with that, thank you. And without any further ado, let me just begin and would like to invite the session chair, Dr. Ravi Trivedi, for his inaugural remarks, and then would request him to introduce our first speaker, Sampada Khopkar from Shimadzu. Over to you, Dr. Ravi. Thank you.
Yeah, thanks, Deepak, for a wonderful detailing about the session so far. And we are very happy to share with our new audience. And I know most of us are associated with APA since long. And this year being a challenging year, but APA came up with a nice idea of having our e-sessions. And so far, it is really turning out as a big success, where audience sitting in home are getting an opportunity to attend sessions for the discovery, for CMC, for regulated bioanalysis and biotransformation. So not taking much time, and as we can see, today's sessions, there will be 3 different sessions. And the first session, we will be starting with the Sampada Koper. And Deepak, you want me to go ahead with her introduction and start, or you want to come back again?
No, please,
Okay.
please proceed further. Yeah.
Yeah. So today's session, the first session is by Sampada Khopkar, and she is currently associated with Shimadzu Analytical Private Limited, and where she is working in the capacity of assistant manager. She has done her post graduation in bioanalytical sciences from University of Mumbai, and working with Shimadzu Analytical India for almost a decade now. And in her increased responsibility, she has catered the need for the spectroscopy, elemental analysis, and which includes FTIR, particle size analyzer, ICP, and ICP-MS. Her work area includes application development and customer support in various fields like pharma, biopharma, clinical area. She is keenly interested in biopharma industry and development of methodologies to cater its workflow. And today her talk title is on Shimadzu's aggregate sizer, where she is going to talk about how this tool helps for evaluating biopharmaceutical aggregation. So without further ado, I invite Sampada Khopkar for this wonderful talk. Over to you, Sampada.
Thank you, Dr. Ravi. Thank you, Dr. Barut, for this nice introduction. I hope my voice is clear and I'm visible also.
Yes.
Very well.
Go ahead, please.
I'll just share my screen. I hope my screen is visible. So can you please confirm once again, sir?
Yes, Sampada, you can go ahead, please.
So good morning, everyone. So today we will present the Shimadzu aggregate sizer, that is the tool for evaluating the biopharmaceutical aggregation. So before going ahead, I would like to just brief you in a short term, like what is protein aggregation. So as we are all biopharmaceutical manufacturers or working with this area, you may aware that the protein aggregation is undesired interaction between the protein and the manufacturing process.
Deepak, are you online?
Yeah,
Interaction, so origin and from a cluster.
Sampada,
Hello.
yes, now you're audible. Actually, we just lost you for a brief this thing. So maybe would you like to start from the starting? I mean, with this slide, at this particular slide, you can start again. Yeah, thank you.
Okay. So just I would like to introduce to you about the protein aggregation. So protein aggregation is a process or is an undesired interaction between the protein monomers. So original purpose of the biopharmaceutical manufacturers was to manufacture these monomers, store them and administrate them to the patient. But the multimer monomers can come together and form a cluster, which is called as the aggregates. As these aggregates become larger in size, they may lose their effectiveness as the drug, but can also cause the adverse side effects, and even they may even results in death. So therefore, the protein aggregation characterization is very much important for all the biopharmaceutical manufacturers during the product lifetime, at least during the product lifetime, at least the ones. So these protein aggregates are classified based on their different mechanism, and those are classified as based on its size, reversibility, its conformation, the chemical modification, and the morphology. One of the factor, or one of the point in which these aggregates are classified is the size. So generally, these based on the size, the aggregates are characterized or those are studied. So protein aggregates are considered as the molecular variant that results from the changes that place in over the period of time or by some chemical actions of the physical factors. So aggregates are characterized or aggregates are of different sizes. They generally range from tens of nanometer to hundreds of micrometer with the different densities. They are generally divided into three ranges, that is the nanometer range, then the subvisible particle, and then the visible particle. So generally the nanometer range covers by the dimers and the tetramers, and basically the subvisible range, which is the most important range for all biopharmaceutical manufacturers, is the from the micron in the submicron and the subvisible range. So what is the significance of this aggregate analysis in the biopharma? So as these protein aggregates can form at any stage of the manufacturing, during the bioprocessing or during the purification or during the product development or the formulation development, during the transportation or the storage. Because of this, these biopharmaceutical products may lose its effectiveness or they may lose its potency, its stability, its quality. So because of this, protein aggregation is a major concern for biopharmaceutical manufacturers as they potentially affect the immunogenicity and pharmacokinetic and pharmacodynamic profiles. Hence, it is important to not only to detect the control and also to minimize the aggregate formation in the final product so that the drug will be or the product will be effective and it will be safe for the use into the patients. So these are the major parameters or the major factors which are affecting the aggregation. So basically these are divided into two categories, that is sequence based, that is it is because of the properties of the protein. The protein has a R group of amino acid. Because of this, they have the properties like hydrophobicity, the net charge, the the secondary structure, and it's the quality or the purity. The second one is the environmental base. The environmental base is due to the environment during the process purification, that is during the upstream or the downstream processing, or during the formulation development, or during the shipment or the transportation. So these are the factors which are important to study for the environmental base. So most of the time, the environmental factors governs, or these are majorly responsible for the aggregate formation, like the protein concentration, the temperature, the stress conditions, also the container closure system. And third effect a third factor foreign substances or any foreign aggregation. So in all these conditions, it is important to study the effect of these different factors on protein aggregation. So these are the few analytical methods which are shown over here for the aggregate measurements. Based on the size of the aggregates, the different analytical techniques or analytical tools are selected for the measurement. If it is in nanometer, then it will be like a SEC or a AUC techniques are selected. When it will be a submicron range, and then it will be QLED technique. If it is in micron range, then it will be a flow imaging system or a light obscuration. And if it is a visible particle, then we can easily find out whether the visible particles or visible aggregates present or not. So if you can see over here, no single method can cover the complete range of quantitative or this measurement of aggregates. So because of this, always the orthogonal techniques or orthogonal methods are used to measure the aggregates. So what is the general workflow for aggregate evaluation in biopharmaceuticals? So generally the biopharmaceutical manufacturers evaluate the aggregates based on its size and the quantity. So that these are the different levels in which those can be evaluated. That is during drug discovery, the researchers find the ways to inhibit the aggregate formation. During formulation development, they can find or they can screen the storage conditions, the use of stabilizers, the use of additives, so that they can reduce this or they can minimize the aggregate formation. And lastly, the quality control chair, where the final product is tested for the aggregate for any aggregate presence of aggregates. So these are the few governing bodies or the regulatory bodies which are important and which governs this aggregate analysis, like the US FDA, ICH guidelines, and European Medicines Agency. So they describe the aggregate analysis in a different biopharmaceutical products or the protein therapy products. So if we look at we can take an example of the FDA requirement, that is US FDA has mentioned the aggregate analysis to be performed in the range of 0.2 micron to 10 micron range, where Shimar's aggregate sizer, that is based on the quantitative laser diffraction method, covers the range from 0.1 to 10 micron range, which completes the submicrometer and a subvisible particle range. So this complete range covered by mentioned by US FDA is covered by the aggregate sizer. So aggregate sizer is a single tool like that.
So Sampada, there seems to be some voice concern, and if you can hear us, Sampada and Deepak.
Yeah, Sampada, can I just, I think everything is clear. It's just that connectivity seems to be a little issue. Sometimes you are lost. So let's do one thing. If you can switch off your video, maybe the audio would be better, and then you can continue.
Yeah, that is what I was about to say.
Okay. So this aggregate sizer can cover the quantitative analysis range from 0.1 micron to 10 micron range. The system is provided with the single laser light source, the single optical system, and the single detection system. Because of this, it can measure the complete range from 0.1 micron to 10 micron seamlessly. It is provided with a violet laser, which measures the small particles very clearly and very accurately. It is provided with a high sense detector, that is ring sensor 2, that is photodiode sensors. So because of this, we can measure these small particles along with that of a larger aggregate size particles. It is with WinSalt Bio software, that is also the 21 CFR Part 11 compliance software, with which we can measure the particle size along with that of a quantitation, that is quantitate the amount of aggregates present in the solution. So the minimum concentration detected level is the 0.5 microgram per ml. So just let me introduce to you how this principle of the laser diffraction method. So this laser diffraction method is based on the scattering by the particles. So whenever the particle is irradiated with a laser beam light, so particle will emit the light in all the direction. So this emitted light is called as the scattered light. So based on the particles based on the particle size, this scattering will depend. So when the particles are larger in size, the light scattered by the particles will be directed in the forward direction. Whereas when the particle size is becoming smaller, the light which is emitted or scattered by the particle will not only be focused on the forward side, but also it can go to the backward side and also the side. So let's just take an
Sampada, are you there?
Yes, sir. Can you hear me?
Yes, I can hear you. Unfortunately, in between, you're getting lost. I mean, not much, but just for a few seconds. So no worries. You can share the screen again, and from that slide you can start. No worries. Yeah, because I can see the connectivity is not that good at your end. The signal is showing yellow. So generally, if it is white, means the connectivity is good. So.
Sir, now is it okay? Can you just confirm once again? I'm so sorry.
No, absolutely no issue. This is a few technical glitches can come anytime, so don't worry about it. Please carry on. Yeah.
So just take a user pointer. Okay. So as the particle size goes on reducing, the particles become smaller and smaller. As you can see over here, the light intensity or the light pattern is changing. So the light pattern is moving towards the outer side. That means it's spreading out. And as particles becoming smaller and smaller, the light is spreading out in not only in a forward direction, but also in the backward direction. So there is a correlation between the particle size and the light intensity distribution. So this correspondence between the particle size, that is particle diameter, and light intensity distribution pattern is very important, or it is basic principle of laser diffraction by which we can measure the particle size. So let me introduce to you the optical system of aggregate sizer. So this is how the optical system of aggregate sizer look like. It's a very simple and very much user-friendly system. So there is a light source which can emit the light. Then there is a collimating lenses. So lens will focus the parallel beam of light onto the particles. The particles are dispersed in a solution in a liquid form into the cell. Then the particles will emit or scatter the light. This scattered light will get focused with the condenser lens onto the detector, that is a WinSalt detector, which is in the forward direction. The particles of a smaller size, which will spread out the light, In that case, there are detectors which are present at the side and the backward side. So with this, we can measure the complete light which is emitted or scattered by the smaller particles and also the larger particles. This is how the detector will look like. So detector can measure this scattered light up to a 60-degree angle, so that whatever light, scattered light falling onto the detector, that is completely getting captured and measured by the detector. So this is called as a wing detector because it looks like a bird's wing. So there are 56 photosensors which are present at the center. Those are called as the ring sensors. And whereas the 11 elements each side of the wing sensor, that is called as the ring sensors. So total it is 78 elements, along with that of this side and back 6. So total it will be 84 elements which are present, photosensors are present, because of which we can measure the aggregates of the accurate size. So this is a aggregate sizes system. What we talk about today is the quantity. It is based on quantitative methods. So it measures the quantitatively the concentration also. So what does quantitative laser diffraction method? So generally, whenever we measure the laser diffraction or whenever we measure the particle size in any particle size analysis system, generally we consider the total population as the 100%. So that is called as the normalized particle size distribution, where we find out the D90, D50, D10 like this. And based on this, we can say that what is the population, the 50% population or 90% population of which size. So this scattered light intensity is proportional to the concentration. As you can see over here, there is a sample A and a sample B. So sample A is having some light intensity distribution. Sample B is having the light intensity distribution. If we add the more sample, the light intensity distribution will go on increasing. So it will depend upon the concentration by which the light intensity will depend. So generally, these light intensity distribution patterns get converted into the particle diameter. So this particle diameter will give us the particle size. So if you can see here, sample A and sample B is showing the similar size or the same size for the particle size distribution. But if we calibrate the system with some standards, that is the polystyrene latex standards, in that case, we can correlate this light intensity to the concentration also with the concentration unit. So with this, we are calibrating this system with the polystyrene latex at the 3 different points, that is at a 0.1 micron, 1 micron, and a 10 micron, because of which we can find out the concentration of the aggregates in terms of microgram per ml or a counts per ml. So it is very easy to get the idea of the size along with that of the particle concentration, the aggregate concentration which is present in the solution. So this is the workflow for the analysis. So this is a single process or one-step process completely happens with the software, that is with the WinCell Bio software. So whenever you disperse the sample into the cell, there will be a light intensity distribution. The light will fall onto the detector, scattered light, and this scattered light then detected by the detector, that is the sensor elements. Based on this light intensity distribution pattern, one can easy to find out whether the particles are smaller or whether the particles are larger in size. And then this light intensity distribution pattern curve converted into the normalized particle distribution data, and then it will be also can be converted into the concentration data. So this complete measurement is a single step or a single process which happens one by one in the particle size distribution analysis. So this is how the aggregate sizer will look like along with that of a temperature controller function. So it is provided with a temperature controller function so that analysis can be performed at a controlled temperature between 20 to 42 degrees Celsius. There are two major accessories which are provided, that is the microcell accessory and the batch cell accessory, along with that of a temperature control water circulator. So with the temperature control water circulator, we can maintain the temperature of the system for the analysis between 20 to 42 degrees Celsius. So analysis can be performed at a constant temperature, and then whatever the changes are happening in the aggregates, those can be studied. So with this aggregate sizer, with the temperature controller system and temperature controller functions, we can measure the effect of temperature onto the aggregation. With, so we can collect a total 200 data points. Measurement can be possible for the collection of this data with a smallest interval of 30 seconds. So this is how the software will look like, in which we can measure, we can see the time remaining or time elapsed, and the cell temperature, whether the stirring is on. So all these conditions can be set, and we can perform this analysis for a single sample even for 7 days. So even if we want to study this kind of a temperature effect onto the aggregation, we can measure for a maximum 7 days for the examination. So this is the batch cell accessory. So it looks, as you can see here, it is look like a cuvette that is generally used in the visible spectrophotometer. So the batch cell contains the solution. Amount 5 ml of the solution is required. So mechanical stress or mechanical stimulus can be provided by the 3 different types of stirrers, that is made up of a PEEK, glass, or the stainless steel, so that we can see the effect of the aggregation of these materials. So this complete accessory is also provided with the temperature controller. So this is how it will look like. So laser light will fall onto the particles. The particles will emit the light, and during this process, the stirring plate will continuously move in vertical motion so that it will create strokes, and the aggregation effect can be studied. The next is the microcell accessory. The advantage of this accessory is this just requires a 125 microliter of the sample. So sample requirement is very less. So you can fill this cell and then clamp it, and then can be fixed into the aggregate sizer. So cleaning is also very easy. You can clean it with any solvent or any buffer solutions or the detergents also. So just take an example for this applications of this aggregate sizer for measurement of aggregates. So first example I would like to show here the effect of stabilizer additives to inhibit this aggregate formation. So we have checked the effect of L-arginine onto the bovine serum albumin when it is measured with the batch cell accessory. So we have prepared a solution of this bovine serum albumin with a trace buffer, and in which we have studied the effect of L-arginine with addition and without addition. As you can see over here, this is the relationship between the measure concentration measurement of the aggregation and the stirring time. So as stirring time goes on increasing, the aggregation starts forming, or aggregates start forming, and aggregation increases. So if we can see over here, when there was no additives added, the aggregation was faster, and the concentration of these aggregates are also higher. When there is an addition of 100 millimolar of L-arginine to this BSA solution, the particle size or the aggregates formation was less. So that means the effect of this such kind of additives or stabilizers can be studied, and then aggregation process can be inhibited. So this is how the the quantitative analysis look like. So without L-arginine, as we can see here, the distribution pattern, how it's like it is spreading from 0.5 to up to going up to 20 micron or a 10 micron range, and then it is spreading out. Whereas when we added the L-arginine, so particle size concentration was very less as compared to that of without L-arginine addition. The second example is the addition of or the effect of different pH levels onto the solution of a biopharma solution. So here we have taken the gamma globulin, which is dispersed in 3 different solutions, that is in purified water, in phthalate buffer, and a phosphate buffer. And then those are analyzed by using the small capacity cell, that is with a microcell. Okay. As we can see over here, the stability of this gamma globulin in purified water is much less as compared to that of a phosphate buffer solution. So in purified water, this gamma globulin is forming a very high amount of aggregation as compared to that of a buffer solution. So in this way, we can study the stability and the stability of the biopharmaceutical products and the effect of pH, and we can select a particular condition for storing them. The next example is the stress study analysis, which we have performed using temperature controller function. So here, again, the gamma globulin, which is used at a concentration of 1 milligram per ml with a PBS as the buffer. This sample is set onto the instrument or set into the cell and kept for a 42 degrees Celsius with a peak stirring plate. So stirring is continuously provided for this analysis, and the effect of temperature has been studied. As we can see over here, as the temperature, as the time is increasing, the aggregate formation has been increased. So the effect of such a aggregation can be studied onto this system. So this is how the cell will look like. So this is the cell which is set inside the instrument for the aggregate analysis, and then continuous stirring mechanism will give you the aggregate formation. So this is how the aggregate formation can be studied over the period of time and with a formation at a different temperature levels. So first, we have studied for 23 degrees Celsius with a different stirring plates, that is with a peak, stainless steel, and the glass. As we can see over here, is the aggregate formation in the range of 2 to 10 micron was higher than we have used the peak as a stirring plate. Whereas when we have used the stainless steel as a stirring plate, the aggregates was higher in concentration from 0.2 to 2 micron measurement range. And this is the aggregate formation time series graph, as we can see over here. So we can see and we can plot this time series graph onto the software, in which we can see the effect of the temperature onto the sample. So we have done a few collaborations with Osaka University and the College Pharma and the University of Tokyo for the free publications, which are there available onto the website or onto the Google, which you can refer for this quantitative laser diffraction method, which are compared with the resonance mass measurement and the flow imaging system and the light obscuration systems. So to summarize, what are the advantages of this aggregate sizer? This aggregate sizer is the best tool for the quantitative analysis of the aggregates across the wide range of particle size, that is a complete subdivisible particle range. It measures the aggregates with a small amount of a sample. The minimum sample amount required is just a 125 microliter. So aggregation can be studied over the period of time in a real-time measurement function. We can study this accelerated testing or the stress studies by using a mechanical stimulus, that is mechanical forces we can apply by using the stirring mechanism. The temperature control function provided so that we can measure the effect of temperature, and also we can see the effect of stress conditions at a constant temperature. The time series changes of aggregation can be confirmed quantitatively, so we can measure this aggregates in concentration form as a counts per ml or as a microgram per ml. And this is the only one technique which can cover the complete subvisible particle range. It is very simple, fast, and easy to operate. So So just thank you for your attention, and you can refer to these two guideline books or the literatures which are available on Shimadzu website for the biopharmaceutical solutions. Thank you.
Thanks, Sampada, and I really appreciate for your wonderful talk, as well as like besides the fact technical glitches, but you covered so well in time. So Sampada will take questions like in panel discussion, and Deepak request you to like share a small token of appreciation in the form of certificate as a speaker for Sampada. So Sampada, kindly accept on behalf of all the like audience as well as like APA committee for your wonderful talk. Thank you,
Thank you
Sampada.
so thank you so much, sir.
Yeah, thank you very much. And before we move to next session, Deepak, Professor Sanjeeva Srivastav, most of us are aware. For those who are not aware, the next talk is by Professor Sanjeev Srivastav. He has obtained his PhD from University of Alberta and postdoc from Harvard Medical School in 2009. And after that, he decided to serve like Indian Institute of Technology, Bombay, and where he is currently working as a full professor. He has established one of the best state of art proteomics facility, which we all know, and where he is having advanced mass spectrometers, microarray SPR setup, and all other required instrument for proteomics. His research on protein biomarkers of infectious disease and brain tumors has resulted into more than 130 publication, 2 books, and over 20 patents filed in his name along with co-authors, including Nature and Cell Press publication. This shows the testimony of work which his lab is carrying out. He has been awarded many national and international awards, including Young Scientific Footstep Awards, Canada, Martha Piper Award for Communications Excellence, Apple Research Technology Supports Award UK, Young Scientist Award from Department of Atomic Energy and Department of Science Technology, India, IIT Bombay's Young Faculty Award, and Professor S.P. Sukhatim, our Excellence in Teaching Award. He is a fellow of Royal Society of Biology, Royal Society of Chemistry, and Royal Society of Medicine. He is visiting professor in Nottingham, Trent University, UK, Arizona State University, USA, Central South University, China, and Asian University Alliance Scholar. So without further ado, I would like to invite Professor Sanjeeva Srivastav. His talk title is High Resolution LC-MS/MS Based Proteomics and FTIR for COVID-19 Prognosis and Therapeutics. Welcome, Professor Sanjeeva Srivastav.
Good morning, Dr. Ravi, and all the organizing team for your kind invitation for this wonderful webinar series. Let me share my screen. Good morning, all the participants. I'm Sanjeev Srivastava from IIT Bombay. I'm going to talk to you about how some of the bioanalytical setup, especially mass spectrometers and FTIR, could be utilized for various applications which you are seeing, but also at the time of pandemic, how we could utilize them for even COVID-19 research. So I come from IIT Bombay, which many of you from India are aware of, that it is one of the top institute in India and also Institute of Eminence from Indian government. I work in Department of Biosciences and Bioengineering, which is primarily focused on the postgraduate education, very strong PhD program. And we have also MTech and MTech in Biomedical Sciences and MSc in Biotechnology programs. So briefly about my talk, I will introduce you to a few of the important technologies related to the proteomics, especially mass spec-based approaches. And then I'll cover about application in area of detection, prognosis, and therapeutics in context of COVID-19 research. So all of you are aware of the Human Genome Project, which led to first draft of the human genome happened in 2001, 2002. And it took almost a decade or more till we could actually get the experimental evidence that those genes actually make proteins in our body. So using mass spectrometry as well as tissue arrays, different groups have tried to provide experimental evidence that for all those genes, almost 20,500 genes, for those we have the proteins do exist in our body. And only 70% of the proteome, or looking at all the proteins, was possible to decipher in 2014 and 15 as a draft human proteome map. Based on the continuous community engagement and the efforts from Human Proteome Organization and the project from Human Proteome Project, only last year, 2020, uh, the large consortium group, including ours at IT Bombay, we were able to produce first high stringency blueprint of the human proteome, which now produced 90.5% of the human proteome with very stringent mass spectrometry based data analysis guideline, and that was published in Nature Communications last year. Still, as you can understand, that we have not reached to 100% protein understanding, still gap with almost 10%. And that gap is known as missing proteome. It means still one need to understand those 9.5% or 10% protein, where they are expressed, what is their function, how we can ensure that, they do exist in our body, and what is the techniques or experimental methods to detect those. So that's where still we have a lot of challenges, which is being taken by the Human Proteome Project and Human Proteome Organization teams. Broadly, when we talk about proteomics, what we are actually looking at, we are looking at analyzing the complex protein mixtures, digesting those to peptides, and then analyzing those peptides in mass spectrometers. There could be different workflow for analyzing that data. For example, data dependent acquisition. When you have large number of peptides coming in the mass spectrometer, you are selecting the most intense precursors and doing fragmentation, then looking at their fragmentation pattern. That is comes under the data dependent acquisition. Or data independent acquisition, when you have all these precursors which are coming in, you are fragmenting all of those and then detecting all the fragments from those. Based on these, there are different type of methods, which I'll briefly talk in the next slide. Also, once you know that which type of proteins are highly interesting in your biological question or clinical question, then you would like to take those forward for the validation using multiple reaction monitoring or parallel reaction monitoring based targeted proteomics concepts. So coming to the data dependent acquisition, especially the label-based methods are very popular nowadays, based on tandem mass tags or isobaric tag for relative and absolute quantification. Imagine a situation when you have, when you are looking at on a cell line, you're looking at some biopharma product or some drug inhibitor, and you want to find out what is the impact of that drug on this particular cell line. So let's say 3 of the samples are controlled and 3 of them are treated with the drug. First, you have extracted the protein, whole proteome from that cell line, let's say MCF7 or HCT or any other cell line. And then you have digested the peptides, obtained the peptides after digestion using trypsin. And each one of those samples separately is now being labeled with these type of tandem mass tag, which binds to the amino terminal sequence of these peptides. These, label reaction comes in different type of kits. Either it could be 6-plex, even it could be 10-plex, or now even 14-plex. So it means you can compare that many states and that many conditions at the same time. So in this case, let's say we have 3 controls and 3 drug-treated cell lines. You have labeled them with different type of tandem mass tag labels. What the number shows here, known as reporter ions, because these reporter ions will be detected in the MSMS level when you fragment those, and that is where you are going to look for the quantification of these peptides. Additionally, one could also look at without labeling and try to measure the abundance of peptide. Idea here is, if your chromatography is reproducible, if your peptides are separating, let's say in 2 hours gradient, at a specific 90 minutes, for example, a peptide with the hydrophobic hydrophilic composition elutes out. In your control condition, same peptide should also elute out in your treated condition. But if there's a change in the abundance of that peptide, that you might see as a part of either the spectral count of the peptide or relative abundance of those peptides. And that's where you can do the label-free measurement. It means without labeling, you are indirectly measuring how much abundance of the peptides are there in those given samples. So these are the kind of different workflow utilized in the field of proteomics: label-based or label-free, data-dependent acquisition. Also, there is a data-independent acquisition where you first make the library, let's say 400 to 1000 M by Z, but separately you take each one of these small windows from, let's say, 400 to 500, 500 to 600 onwards, and generate high resolution library from those, and that library will be utilized for the subsequent overlaying the spectra and obtaining the high resolution MSMS data set. A popular method based on this is, comes from the SWAP technology, from the triple TOF instrument. Also with the Orbitrap, not DIA is possible using the Orbitrap different configurations. When you want to validate your targets, now you have identified a given biomarker or proteins of interest, then you are going to focus only on selected peptides. You don't have to look at, again, those thousands to millions of peptides. And that's where targeted proteomics-based workflow based on triple quad mass spectrometers comes into the play. And MRM multiple reaction monitoring based on a triple quad configuration depicts that, like, let's say you have many peptides which is coming flying inside the from the after ionization, they are coming to the mass analyzers. And in the first quadrupole, you're only selecting the peptides of your interest, which is getting fragmented in the collision cell, followed by in the second quadrupole, you're going to do further fragmentation of those. For each one of these precursor and their fragment pattern, you are generating the transitions, and these transitions are monitored based on the time and the relative intensity scale. This is where you can develop the specific selected reaction monitoring or the SRMS based on the targeted peptides. More recently, there has been approach of PRM or parallel reaction monitoring, which primarily looks at the possibility of fragmenting all of these precursors and then separating them in Orbitrap and looking at the entire peptides and their quantification. Slightly more ambitious, but both are the possible approaches of looking at targeted proteomics-based assay development. Some of these things I will talk to you in the subsequent part, in the application part, and I'm sure I don't have to introduce you the terminology and the concepts again. So we at IIT Bombay have set up this national facility of proteomics, mass spectrometry, based proteomics research and training facility, which is equipped with variety of high resolution mass spectrometers and triple quad mass spectrometers for various applications from discovery to targeted based analysis. Of course, this is being a national facility, anyone is invited to use, and it's heavily used by various academic institutes as well as different industry sectors. You are also welcome to contact us and join us for any type of your analysis. Coming to some of the biopharma applications as a part of our facility, we also provide various applications from intact mass analysis, reduced mass analysis, peptide mapping, PTM analysis, which is quite challenging, but various type of biopharma applications are based on those. I'll just brief you and give you some little bit glimpses of some of these data sets from different industrial projects before I move on to my application talk for today. So again, looking at peptide mapping, just showing you some VSA-based data, and also looking at antibody-based peptide mapping for the heavy and light chains. Different type of glycosylated forms can be detected using these high-resolution mass spectrometers. One could utilize different type of fragmentation patterns from the high collision density to the electron transfer dissociation, or the combination of those with the ETD and HCD together. All of these really helps to increase the coverage of these intact protein and the antibodies, especially data shown here for carbonic anhydrase. And then also we look for the various type of native protein characterization. Different industries comes with different type of interesting problems, and we try to look together that how best we can provide data, in this case with another industry partner, and more of the biosimilar peptide characterization from another industrial project. And we also look at a lot of biosimilar characterization of peptides. Again, looking at how similar, your innovators, molecules are, and look at those biosimilar characterizations. So again, I'm not revealing any data or any insight from these projects, but just giving you flavor that variety of things can be done from the different type of industrial projects, and also sometime, even challenging samples like FFP based, these tissue blocks to take and do certain type of discovery and targeted proteomics analysis for different type of challenging problems, especially cancer, also being taken forward. So now coming to the, you have got the glimpse of proteomics and mass spec and possible applications in different sectors. I'll now focus on a recent work which we did during the pandemic time. And that's where that was very challenging because every operation was quite, brought the logistics as well as the technical challenges without knowing what we are going to look into. And that's I'm going to show you a little bit of story about what work we did using these technologies. So again, the entire operation was under the biosafety considerations with all the safety and issues of how to protect people who are working on these projects. We collaborated with Kasturba Hospital and Jaslok Hospital in Mumbai for this project, where the samples were initially collected in these hospitals, inactivated for virals, as well as the initial sample preparation. We sent our trained teams to work in the hospital to prepare the sample directly over there. And then only the peptides which were clean and dried, those were brought into our setup for the next set of mass spectrometry and other type of analysis. So of course, it was entirely challenging project, but we tried to look into different problems, in which way we can try to provide some new insight with the as a supporting thing with the ongoing other technologies. So as you're aware that any patient who is being tested for COVID-19, their swab sample, nasopharyngeal or otopharyngeal swab sample, that is being collected, and that is being tested with the real-time PCR, which provides some CT value, threshold cycle. And lesser the CT value, then you assume that there is a more high viral load. So whatever the left sample from the swab sample, which was in the VTM, viral transport medium, that was available to us to see, can we look at the peptides obtained from these patients and look for what kind of virus we can detect, or can we look at the host responses from these samples. So again, we had never worked on the swab sample, and especially with the interference coming from the VTM. So we thought, can we look into, in some way, can we look at possible extraction methods, and in which way we can try to enrich the viral peptides. So we are talking about the human respiratory specimen right now, which should have, if there is a viral infection, some viral protein should be there. And also there is a lot of host protein going to be there, because coming from human sample. So looking at this, we tried different extraction methods from ethanol, isopropanol, acetone, tried digesting those protein to peptide forms, ran them in the Orbitrap fusion instrument to get the very deep proteome coverage, and then selected peptides were taken forward for the SRM-based analysis. So when we look at these patient samples, we were able to get several viral peptides, which is very interesting to see that, from the each extraction method, we could get several unique peptides coming from SARS-CoV-2. The data was searched against SARS-CoV-2 database to make it very unique only for the SARS-CoV-2 virus, which is responsible for COVID-19. But then we thought, why don't we take the pool of these peptides, which will we have a highest number of viral peptides enriched, and use that sample for developing our further SRM-based test, which can be taken forward. So we were having proteins from the spike protein and nucleoprotein, some of the even replicase proteins. First of all, we wanted to see, can we detect these? How low we can go with the triple quad? We also generated some synthetic peptides for developing this assay. And again, when you do these kind of test development, there is a need for looking at a lot of standards and your how well you can try to be confident about your reproducibility in the assay. So again, looking at, we had some of the heavy level synthetic peptides which were used to spiked in to look at reproducibility across different samples, different runs. And only after ensuring those, then we developed this MRM based test, which was based on 11 peptides. So we are quite conservative, rather than looking at just 2-3 peptides, which one could screen in a very short gradient. We went for almost 8 minutes long gradient, which could look for 11 unique peptides of SARS-CoV-2. And based on this, then of course we had a much higher confidence, because peptides are coming from a spike protein, nucleoprotein, replicase polyprotein. Even if some peptides are missing in some patient, but at least you have many other peptides, which will overall give you confidence that it is actually COVID positive or COVID negative. And then we could also see, some correlation based on the CT values versus what we could see with our intensities of the peptides coming from triple quadruple SRMSA. Further, since these samples are coming from the host human, of course, doing the mass spec based test for viral detection may not be the easiest thing to do for the clinics, that we understand. And of course, real-time PCR is available everywhere and easy to deploy in the clinics, whereas mass specs are not so much available. So that was a good test which we could show here, but still we understood the challenges of implementing it to the clinics. Nevertheless, we thought, since we have these sample which also contains the human protein, why not we also look at the host responses? So in this case, now we also look at the plasma proteins as well as the swab proteins, and we search against human databases, and we try to look at what kind of human protein we can see in response to the COVID-19 infection, and are there any changes in negative versus positive or within the mild versus severe group of patients. So now I'm showing you the host data, against the human proteome database, which we searched. And now what you see here, also very interestingly, the negative group is well segregated, whereas the positive group along with the recovered group, they are still quite a bit mapping. So this is very interesting and slightly puzzled, observation for us, that many individuals who have been now recovered from the COVID-19, which were positive earlier, but now real-time PCR showed them they are negative, but still they actually became part of the positive group. So it means still, while they have just recovered, still there's some viral trace or viral peptides can be seen, and that shows the sensitivity of the mass spectrometer. So when we went back to the clinician that many of the negatives are coming as a part of the positive, we are worried what is going on. And then they looked at back the history of these samples, and they found out these are the recovered individuals. They're quite happy to note that these are already positive sample, but just recovered. It means the mass spec is much sensitive to detect still the low burden which is still available. So now that shows you different type of analysis, how that can be powerful, and how mass spec and proteomics could be very sensitive. So we looked at different comparison of COVID-19 patients, looking at even the mild versus severe group of patients. And of course, the patients will have a lot of heterogeneity, which you can see from the intensity of the protein, but still the good patterns could be observed in the mild versus severe group of patients. And that was quite encouraging for us to then look at where these proteins are changing in the COVID positive patients. And we could actually map all the proteins coming from our data, because now we are talking about thousands of proteins which we have identified using mass spectrometers, and looking at most significant proteins where they are mapping in the biological pathways. And we could see that neutrophil degranulation pathways, some of the platelet degranulation, as well as translational machinery, many of these pathways are quite perturbed based on the COVID-19 infection with SARS-CoV-2. And that's where we thought to put more emphasis as we want to go forward, looking at some possible therapeutics. So while I'll come to the therapeutic part a little later, but coming to the prognostic part, looking at the patient response, idea was that detection is possible using mass spectrometer, but that may not be clinically applicable so much because of the challenges involved in doing the detection. But currently, like when the patient comes as a COVID positive, everybody has apprehension that, are they going to be severe or they're going to just quickly recover, right? So that's where we thought, can we look at the host response of mild and the severe group of patient and see, can we find any pattern emerging that how the proteins are changing inside based on the viral infection in the mild or in the severe group of patient. And similar kind of work which we have already done recently on the malaria project, when we looked at Plasmodium falciparum and Plasmodium vivax infected individuals, how some of them are mild and some of them are severe, and what kind of patterns could be observed. So based on that work, we had published last year only in Nature Communication Biology this work, which showed different type of falciparum malaria, severe anemia, cerebral malaria, other type of complications, how they're different than the non-severe falciparum. And we also did the same analysis in the vivax as well. And that was quite encouraging. It was just before the pandemic. And we were quite confident that this, the patient's blood samples, plasma sample, is very good reflection of looking at these kind of changes. So we thought, why not we try to apply the same strategy for the COVID-19 as well. Although we, fortunately, during the lockdown time, we had all the reagents available for depletion of the plasma and all the processing for at least 200 or more patients, because everything, whatever you have available, only you can rely on that. So based on this, then we recruited patients of the negative and positive groups, non-severe and severe group, and collected the blood and plasma samples from these individuals, depleted the plasma for the abundant proteins, and then we took them forward for the mass spectrometry analysis to find out which are all proteins present in the plasma sample, and then can we later on do the level-free quantification to compare any pattern emerging from the mild and the severe group of patients. And after doing this type of all different software analysis, finally we were able to select few peptides to develop the SRM assays, and few leads were taken forward for the potential drug repurposing aspect. So some proteins I'm showing you here, which are also quite known clinical markers. So while during the pandemic time, there was no test like this available, but doctors were doing some indirect tests using other biochemical assays or the ELISA test, looking at the patient responses for the ferritin protein, interleukin 6 protein, some of the D-dimer, these kind of proteins, which we were very happy to see we could also detect in our own sample. But we could also find out there are a set of protein which is not reported, which is also looking very interesting. And few of those, especially apolipo B protein and fibrinogen gamma chain protein, turned out to be very, very interesting for us. So all of these things actually show to us that there's a good promise of using the mass spec approach to find out some new targets and new leads, which can be taken forward for the developing MRM assays. So now I'm just showing you a couple of quick slides to show, as I already explained you how MRM assays work, that we were able to now look at both swab sample and the plasma samples and try to specifically look at which proteins, peptides of our interest. In this case, interleukin 6, lactate dehydrogenase is already known for the clinical markers, as well as C-reactive protein and AST. We could see very clearly that negative and positive, we could measure differences in the MRM-based test. But what came very interesting based on our plasma data, which was not obtained from any other technology, is a set of proteins, especially angiotensinogen, apolipo B protein, serpin proteins, fibrinogen gamma chain protein, all of them, and the serpin other family G1 protein, all of these proteins showed that there is a very clear difference one could observe from the non-severe and severe group of patients. And that's where we thought to put our maximum bet of developing the panel of these markers to look for the severity progression of the disease. So along with this, since we had the deep proteome data, we could also dig deeper into looking at the host protein responses, and we could see from plasma, there's a complement activation pathways, leukocyte activation pathway, which is quite a bit changing in the blood samples. And of course, as you are aware of, because of the SARS-CoV-2 viral infection, even after virus goes off after 4 or 5 days, but because of the viral damage in the immune response, so much cytokines are being released, cytokine burst happens, and those cytokine storms which you have heard is actually then triggering a lot of the changes in different pathways, and the immune system gets perturbed. And that's where we could see many of these pathways are getting quite a bit modulated. So we selected some of these key pathways, and idea was, can we look at some of the drug targets which could be inhibiting maximum of these proteins which have been identified from our severity markers, and loratadine and UCPH101 turned out to be some interesting protein to dog these targets. But selinexor actually came out to be quite interesting, which was able to inhibit 6 proteins which are responsible for severity. And some of these work is still at the comparison level, but need to be further validated at the cell line based models to test out their potential. But by now we have already seen a couple of other studies have also reported selinexor as one of the interesting target for inhibiting toward the COVID-19 severity. Additionally, since we were looking at the changes happening in the mild and severe group of patients of COVID-19, we thought, can we also look at some very quick clinical assay development, because we see there is a change in the plasma already. So we use this system from Agilent, the AT-FTIR instrument, and try to look at the FTIR spectra. Is there any difference in the severe and non-severe group of individuals which we can measure, unlike mass spec, which gives you the exact peptide sequences? Here you don't have that specific information, but here you can just see like in which wavelength regions you have some changes happening, and that could be coming from either the amide bond changes or coming from the sugar residue changes. So any of those changes might be reflected in the non-severe and severe group of patients. This work was done in collaboration with Agilent as well as QIMR Australian teams. We looked at the plasma samples and we were very happy to see that there are some spectral differences which were being modeled with the Australian team. And with a lot of rigorous testing and blinded testing, we could reach quite a bit of 94% or so sensitivity. And that actually turned out to be one of the very interesting way of even rapid detection for the severity prognosis. Of course, more authentic information can come from mass spec, but a quick segregation can be done using this kind of point of care devices, which are very small and handy devices to be deployed. This work was highlighted in several media and a lot of places. More recently, we have also looked at the longitudinal sample. It means the same individuals have progressed from the mild to the severe group of patient. As you may understand, it's quite challenging sample to obtain, and still we were able to get almost 25 or so patient to look for these transitions, and some of the markers are confirmed. Also, we could see that leukocyte activation pathway emerged as one of the key pathways which is changing as the disease is getting progressed. This became the cover page of the Journal of Proteome Research in the last week edition. Just to summarize from this entire work, we were able to utilize the MRM-based assays from the swab. We could also look at the prognostic-based markers. We could look at some of the novel key pathways and some of the drug candidates for the repurposing aspect. We were invited by Nature to give the webinar to showcase some of the applications coming from mass spec-based approaches to use for the SARS-CoV-2 detection and prognosis. The work was generously funded by SERV at the time of first wave of COVID-19 pandemic immediately. So we're very thankful for the generous and quick support. So was IIT Bombay Seed Fund. And eventually we have more projects funded by Department of Biotechnology and Indo-US Science Technology Forum working in this area. I'm not acknowledging other projects that are funding today. A huge number of researchers and the scientists, clinicians were involved in this particular work. So I'd like to acknowledge all of them. Due to the time shortage, I'm not going to name each one of them individually right now. Before I close, I'd like to announce that we are also quite committed to provide training and more educational component to the society. We will be conducting a free 10 days online workshop in November. Some of you who are interested to learn more about proteomics could keep an eye on some of the notification which will be sending soon, and are welcome to join this initiative. Thank you everyone for joining for today's webinar, and I'll be happy to take any questions live or on the email which is shown here. Thank you.
Yeah, Professor Sanjeeva, as expected, excellent talk, and it was well covered, knowing the very current topic. So very useful. If you have time, maybe a quick question, how about COVID versus other viral infection, let's say dengue or associated one, can it be differentiated, or that was not seen in this research?
Right. So that's actually very interesting. We are doing two different projects to look at how to look at similar type of febrile infection. And the Indo-US Science Technology Forum project, which I just mentioned, acknowledged, with IIT Bombay and Arizona State University has been funded. That's where we are looking at multiplexing test development to look at different type of flaviviruses, how to distinguish those, including Zika, chikungunya, dengue, as well as the COVID-19, because all of them give the similar responses, and look at how best one could segregate those. Even at the mass spec level, we could see that unique peptides can be detected using SRM assay, but to make it more clinically applicable test, we are working with the ASU US team to do multiplex test development using a MISPA technology, a novel technology.
Okay, thank you. Just last question, and remaining we will send through email.
So, thank you. If I may interrupt, Dr. Sanjeev, thanks for a wonderful talk. Can I just request you to press the stop sharing so that we can highlight your video?
Yes. And thank meanwhile, you. By the time Deepak is sharing, so you have mentioned specifically PRM mode in Arbitrap. So were those techniques optimized, or there are other scan mode in Arbitrap which can be used like DDMS2, etc.?
Right. See, I think when we are looking at the deep proteome analysis, or the first, the entire proteome coverage, then of course DDMS2 will be useful. But when you are looking at specific peptide detection, then you have to develop the test for specific peptides only. that's where MRM or PRM comes more important for the the assay development. And PRM, as I said, like it's more ambitious right now because you can assume to quantify every peptide obtained from the entire complex proteome. That is actually working okay, but we feel still much more confident on the MRM assay because that's where you have very much control about your assay. You can take synthetic peptides, generate specifically, where the retention time will be, what is the exact, LOQ, LOD you can obtain from those. And then you can specifically look at the few peptides of interest. Because for the clinical assay development, you don't want to have all the proteins and all the peptides to be screened. Hardly you are going to look for maybe 4 or 5 proteins and maybe 8 or 10 peptides. That's it. MRM is much more powerful and much more stringent in some way. But if the goal was to look at potentially screening for all the biomarkers, then I think PRM will be useful. But again, that is still not reaching to that level of clinical confidence as what we have with the MRM.
So in MRM, it was a unit resolution must, Professor Sanjeev?
Yes.
Okay, thank you. So I will not go further, knowing that time is restricted for you and you have to go for another engagement. So thanks a lot again. And as a small token of appreciation, here
Can I
is
just take a question from a chat? I think one of the participants had asked, especially, the comparison of HRMS from different technologies. I
please.
must say that, okay, various technologies have their own advantages. Just because we are using a specific configuration does not mean everything can be done on the same instrument only. It all depends on your application. And that's where I think, one need to, in the mass spec, you need to be very choosy what application you want to do. And accordingly, you can select the configurations. If the idea is to look for the deep proteome coverage, then Orbitrap definitely comes on the top. If idea is to look at selected peptides, then you have all possibilities to explore from all the vendors. If idea is to look at other applications, then might be triple top other technologies are equally competent. So that's where I'm not going to give you any specific name per se. Every technology brings some novelty, and you have to choose as per application. Thank you.
Thanks, Professor Sanjeeva. So Deepak, can you please display certificate? Yeah, so here is a e-certificate, and thanks again for agreeing and presenting this nice talk. Thank you very much, Professor Sanjeeva.
Thank you. Thanks to organizers and all the participants. Thank you very much.
Thank you.
And thank you, Dr. Sanjeeva. And on behalf of organizing committee, would like to mention that hopefully, looking at the scenario, next year we'll be having an in-person meeting. So we would really like to have you in person to deliver a talk. In advance, we just wanted to invite you and thank you for a wonderful talk. I'm sure there's so many questions. We will come back to you with those questions over your email, and then looking forward to connect with you again soon next year. Thank you.
Thank you so much for nice comments. Thank you.
Thank you, Deepak. Thank you, Professor Sanjeeva. So before moving to next talk, now this is our third and final talk, and thank you very much for staying with us. The next talk is by Rene Vink, and thanks for staying. And this talk title is on solution for today's challenging drug discovery pipeline and NTT technologies. And Rene Vink is currently with the Nanotemper, Japan. And Dr. Rene conducted his PhD and postdoc on molecular developmental genetics with focus on peptide and hormone signaling network in cell-to-cell communication processes prior to joining Nanotemper, Japan. He is a senior application specialist, currently expanding the outreach of Nanotemper in Japan by working with the local pharmaceutical industry, startups, and a wide variety of academic labs to help them succeeding with their projects in protein science and molecular interaction. So I will not take more time, and I would like to invite Rene Vink for his talk. Please, Rene Vink.
Thank you, Mr. Triveda, for the very nice introduction. Can you hear me?
Yes, it is very clear, and presentation is also visible. You can put it in presentation mode. Yeah, all set.
Okay.
Yeah, and Rene, very quickly, if you can switch on your video so we can.
My video as well. 1 second. Where do I do this here? No, and now I'm visible, I believe.
Yes, you are very much visible. Please proceed. Yeah.
Okay. Yeah, thank you very much for the invitation. It's a huge pleasure to talk here about our screening unit, Dianthus. Yeah, I'm sitting in Japan. We opened an office here 2 years ago. So basically I opened it with all our equipment and I'm growing our business in Japan. But I want to focus your attention on our demo lab and office in India, which is more important. And my colleague Chief is running it, who's also attending the seminar. In case you have further questions, we can discuss this, of course, here, but I recommend you to note down this email address or visit our homepage to connect directly to your local office. Yeah. The title of my talk is Tackle All Your Molecular Interaction Screening Projects with Dianthus. So it will be about ligand screenings in single dose and affinity screenings, high throughput, but not HDS, also not hundreds, thousands of ligands, but more like hit confirmation, validation, lower numbers. I will show you some numbers. Okay, so that's how the instrument looks like. It's like a meter wide and half a meter deep, 30 kilos. And that is all you need. So more is not required except maybe a robotic arm, which I will also show later. Yeah, to start off, I want to show you that this technology, also our technologies are established. So we are not new on the market. We are like 13 years old now and very established in Europe and in the US. In Japan, we're growing a lot as well. And Chief, it would be so nice if you could talk a few words as well. In India, it's also very popular and going very well for us. Yeah, so where do we find the Dianthus? I show here like drug discovery pipeline, and we see the application of Dianthus in hit ID, hit screening, and hit validation. So the Dianthus works perfectly on its own, but I mean, it's out of question that there are other methods which are established and working very well for HDS screening, for example, SBR. Dianthus is a perfect complement for these technologies. And I would like to recommend you to consider such a complement, for example, to find hits of low molecular weight changes. So that's one advantage, for example, which Dianthus has to other methods that is completely mass independent. I will show you some more advantages in a bit. Okay, so here's a rough overview of the entire talk so that you know everything in advance. So the Dianthus screens in well plates, directly in plates. I will show you a picture on the next slide. One plate carries 384 wells. You can measure N1 or N2 as you like. And we can measure the entire plate in 30 minutes. And then you can turn over and calculate and upscale how much you need. The measurements and the samples are in solution. So that's the major difference, for example, to SBR. Your samples will be free floating in the buffer of your choice. So you can also use any kind of buffer, also detergents are fine. And the only thing you need is small dye, which labels the protein. I will show you to this also more information. The technology is mass independent, and you can also measure any types of molecules. You can go for DNA, you can go for proteins, peptides, you can do viruses, fragments, ions, anything you like. Okay, buffer independent, we said. Affinity-wise, you can screen from 1 picomolar up to several millimolar. In my talk, I will show you a very short project on a fragment screening where the affinities are very low in millimolar range. And there's another very nice application for Dianthus where it can back up other technologies because it's very good for weak binders. Okay, yeah, plate-based. That means, yeah, no fluidics are involved and the instrument requires no maintenance and no equilibration. The only thing you do, you use a plate and you put it right in here. The Dianthus measures it and afterwards you either store it or dispose it. So there's really nothing you can break on this instrument or you have to clean. Yeah, the plates, I took this photo, look like this. You see some welds I closed off with a lid, with a sealing foil. You don't have to do this. You can screen an entire plate without closing it. But if you want to close, if you have a large screening of like 50, 100 plates, you can close them. Then you have zero evaporation. You can put them in a plate hotel. You can apply a certain temperature to it, keep it there. And once it's ready, it can go in and you will have no artifacts because of possible evaporation. You see here on the side of these plates, there's a code which the dentist can read. And we use this code to store in information of where and which wells you already loaded something and what, and which name the plates have. So if you have large screenings and you want to refer positive, negative controls across different plates in a larger screening, this will help you to organize this in our software. One data point requires 20 microliter of sample. And if you have a target protein, we need 5 nanomolar up to 50 nanomolar. If you measure in picomolar range, with picomolar affinities, you typically just use like 5 nanomolar of your target. And if you go up to millimolar or micromolar interactions, I normally use like 25 nanomolar. And in some cases, when labeling is very difficult, maybe you have to go a little bit higher to 50 nanomolar. Okay. Yeah, the welds are all coated. So these plates we distribute from NanoChamber. You see also our logo here that avoids absorption of the samples. You have no evaporation because we can close them, and of course no maintenance. So that is the only consumable that you need to run these experiments, besides the buffers and your samples and a die. So here is that's a picture we took in Munich in our headquarters in Germany. Here's one dianthus, and they stacked another one on top. I don't know why they did this, maybe to upscale throughput, I believe. So, and here is an example of how we can automate it. They used a precise automation robotic arm from Cobot. And here you see a plate hotel. So it could grip from here, insert it in there. And you can also put a centrifuge next to it, which is automated. And then it's fully automated. Several of our customers integrated this already. It is very easy to do. Okay, so here is a typical workflow of a screening approach. And I want to show you where we can do what in the next slides. So we have as a development in the beginning, we only look at our target protein. Does it aggregate? Yes, no. Is the fluorescence okay to screen? Yes, no. And is the data of our positive control reproducible? So we look at these three things and we optimize maybe a little bit the buffer, we change it or we spin down. And that's all you basically have to do. That's kind of out of the box solution. You can do single dose screening. Let's say you come from an HDS screening and you have 10,000 hits, something like this. And you want to confirm them. You can do single dose screening maybe in duplicate or in unicate. Here in yellow, I show positive controls, and in green, we put a negative control in every row. That's a typical design of the plate layouts that we use. You can also do affinity screening. So here with the gradient is shown a ligand concentration range, a high concentration to a low concentration in a 1-to-1 dilution series. And you can screen in 8 points or in 16 or 24 as you like. Yeah, so that's what you can do. And then you would go to either lead optimization, or you would take a closer look into, let's say, potential hits, which maybe show a little bit aggregation or not 100% reproducible, something like this. Yeah, and to give you some numbers, you can screen 8,300 compounds in duplicate in 24 hours. That's walk away time. I'll switch it on and you get 8,300 compounds in duplicate measured for a single dose screening. You can do 1,500 KDs in 24 hours as well. Of course, it's all flexible. So now I would like to, hello, I think I hear kitchen. Okay, I will show you now how the data looks like, and then I will show you the mini screening that we have done in collaboration with a partner. So here's the nomenclature. We always have a protein that will be labeled with a red small die, and that's we call the target. And we have a ligand that can be any kind of molecule, can also be another protein or a microRNA, whatever you like. And it's called the ligand. So how do we label? So we have kits that we offer. Of course, you can also use your own methods if you have something established, or you can use fusion proteins as well. Most popular is lysine labeling, where we label with a red dye a surface lysine of a protein. We label in ratio 1 dye per protein. And that is the same labeling chemistry as it is used for surface immobilization of SPR. Then you can also reduce cysteine bonds and label here. That is less common because you would maybe destabilize your protein and change the abilities to bind. So I'm not a fan. You can also use a snap tag, which is very good for some targets. They only work with a snap tag somehow, but it's a very good solution to try it out. And very popular is his tag labeling. Many proteins are purified with a his tag. It carries in 6 histidines, and we have a dye that specifically binds to these 6 histidines with nickel binding in low nanomolar range. And yeah, that works very well. So now I would like to talk about where the data comes from and how it looks like. So what influences the fluorescence that we measure? We have here a target protein. We have labeled it with a small dye. And 2 situations can influence the fluorescence properties of this dye. One situation is a ligand binds, therefore in close proximity to this dye, and changes the chemical environment, so the charges surrounding the dye. And the second thing is, if you have, for example, a longer receptor, maybe a transmembrane receptor that's quite long, if a ligand binds here, it might be too far away from the dye, but we have a conformational change of the receptor. This redistributes the charges inside of the protein and also in closer proximity to the dye. And that is enough to sense the change, and that we can use to measure interaction. So it's a very sensitive method, much more sensitive than plate readers. So that is not comparable. We apply a second effect that we use for measurement. So we have here a protein that is labeled with a dye, and it's free floating in our well. And we now increase mildly the temperature in the center of the solution, where we also measure the fluorescence. And if we increase the temperature of a dye, it's a normal biophysical property that the dye fluorescence will decrease. You see here it's much brighter. So it's a normal effect that we always see. And that's what we measure. Here's the raw data depicted. We have here on this axis the fluorescence intensity, and on this axis the measurement time. That is 3 to 5 seconds, what you see here. So we measure first just the fluorescence in the well. Then we have a value that we will normalize for all ligands. And then we increase the temperature from here mildly continuously. And therefore, the fluorescence of the dye drops off. And then we repeat the same measurement in the presence of a ligand. If the ligand is not binding and stays in distance, we get exactly the same curve. And in case it binds and maybe it changes conformation, something like this, we get a curve that is different to the curve without ligand. And we only look for the difference of these curves. So it can go up, can go down, can have all kinds of changes. But if you have a difference, we call this trig signal amplitude. And trig stands for temperature-related intensity change. So we just call it trig signal amplitude. If you have a signal to noise that's large enough, then yeah, we can conclude on binding directly. So I show you the raw data now, a plot here on the right side. We have here the case of non-binding. Then we have an F-norm value, which is a value from this axis here. We have a triplicate, so the noise level is relatively small. And then we repeat the assay here together with the ligand, and we get basically the positive control in triplicate. And the noise level is large enough to conclude on binding. And for screening, I will show you in the next slides, we always see this pink line. And that indicates our chosen tolerance level of the negative control. So we make it always like negative control, what's the average? And then we multiply it normally times 3, and then we have a good value to exclude negative ligands, so noise of negative ligands. Everything which is outside of this pink bar would indicate a hit. And of course, then we would also look at the sample quality to decide, okay, is it now an outlier? Is it reproducible? Is it aggregation? Something like this. Can we trust this hit, yes or no? So that's an advantage of the technology, that we see raw data and quality data of our samples in solution live in front of us. So we can very nicely and how to say it in English, we can avoid false positives, which can be very costly if you don't. So here you see the raw data in duplicate of a single dose screening hit, let's say. In green is negative, and in blue, there's a ligand that binds to our target. And we can show this data in this fashion here. So basically, we compare these curves at any time, any on time of the measurement. Then we can plot this in this fashion. So, and here, again, so we ran a full plate in this collaboration that I will show you in a second. And they ran, then you can run 8,300 compounds in duplicate in 24 hours, or 16,600 single data points. So what is now, how do we get now an affinity? Here we have only the ligand, only the target basically, and 0% ligand. And here we have the highest concentration of the ligand to saturate binding. What we do is we run a 1-to-1 dilution series of ligand. So we will run this experiment again with half the concentration of ligands and half the concentration and so on, till we reach very low concentrations. And that is shown here. Here we have a high concentration of ligand and here an extremely low. And the points in between is the 1-to-1 dilution. And if we have then, let's say from here in this direction, more and more saturation, the curve will change into this direction. At some point, the sample is saturated, and therefore it doesn't matter how high you go with your ligand, it will stay in the saturated form, in the end form. And we can plot these data in this fashion here on the right side. You see on this axis the ligand concentration. So the ligand concentration for each data point. Here we have low concentrations, here we have high concentrations. You see here the protein, the target, is not bound by ligand. And when we increase the ligand concentration, we get more and more saturation until we reach full saturation. And the turning point of this curve, indicated here, will tell us, okay, this is the KD of this interaction. So it's a very simple process to come to a KD. And in case the curve comes up later, we have a weaker binding. In case it comes up earlier, we have a stronger binding. And in case it does not saturate, maybe we only have a very weak binder, and we cannot, we don't have enough ligand available to saturate. We get all data points from here to here maybe. Then we can say, okay, the KD is at least equal this concentration or higher. And that is very variable, for example, for fragment screenings, to identify these hits and then go forward with optimization. Yeah, here again, just to remind, to give some numbers, you can screen a full plate of affinities within 24 hours. These are multiple plates, of course, 1,500 KDs. Okay, now I want to very shortly talk about how we optimize these kind of assays before we start the screening. So yeah, there's not much to do. Basically, we spin down to get rid of possible aggregation. That's always a very good choice to do. We spin down the stocks and then we start working with it. And then you remember we have the amplitude of negative and positive. And depending on which buffer you're using, this can be smaller and taller. And we offer a deep well plate, it's depicted here, with all kinds of buffers that turn out working well in trick assays. And here's a little bit more detail what kind of buffers we loaded in there. And what you can do is you can run, for example, the buffers that you like. You run 2 wells, just the target, and 2 wells, target plus a high concentration of ligand, and then do it in the buffers that you are interested in. And then you get the following results. You get a heat map. And the darker these experiments are, the better the signal quality, so the quality of your data is. So this one is an optimal buffer, and white is a very bad buffer. There might be aggregation or no amplitude. Here I have an example. For example, if we click on this buffer, we see this result. We see a relatively small amplitude, but it's acceptable. We see no aggregation. Aggregation would be if we have outliers, if the curves go in different directions or cross each other. And if you pick this buffer here, you see the amplitude is significantly larger. And that is more optimal for a screening in case, to separate the signal to noise of negative to positive. So you select the buffer, maybe 3 buffers that you like, which return the best result, and then you go forward with your screening. The second thing what you can also do is to optimize a positive control before you go forward with maybe an expensive screening. You want to make sure that your protein is not denaturated, not partially denaturated. You want to know, is it very stable, or maybe it needs more flexibility? So you want it a bit more unstable. And you also want to know maybe, okay, I have a positive control protein and a ligand. When I mix them together, normally ligands bind, they maybe change the conformation of the target protein and also the stability. And you can measure this very quickly with another instrument that we developed. It's called Tycho NT6. It can tell you if your ligand bound and if the conformation of your target changed. So that's a label-free nano DSF solution. So you might know DSF that uses a dye, and this one here is label-free. So what we do is we load 10 microliter of our protein that can be extremely low concentrated, like 5 micrograms per milliliter. And we expose it inside of the instrument to a thermal, to high temperatures. So we ramp up the temperature from 30 degrees to 90 degrees. At some point, your protein will unfold. And then we have an inflection of this unfolding curve. And that will tell you the stability of your protein. And now, here's the experiment to the screening. In gray, we use only, in blue, sorry, we only use the target protein. We unfolded it. And we see it has a stability of about 50, 53 degrees. And then we repeated this assay, actually in the same run, together with a ligand that is supposed to bind. In this case, it's an inhibitor. And we see this curve here in gray. And first we see the stability, the inflection here, change to a higher temperature. Therefore, we have stabilization upon binding. So it strongly indicates already binding. And the second thing, which we can see here, which is not possible if you use the cypodye, for example, you see the initial ratio on this axis is also changed. And this indicates this upon binding of the ligand, the conformation of the protein changed. So you have 2 very good parameters to conclude, okay, we have now a good amplitude for our trick measurements, and our positive control will work very nicely. If you have new compounds, maybe low throughput, something like this, before I start with assay optimization or trying different ligands, I always run this kind of label-free thermal shift assay to know what to expect. It saved me sometimes several days of work of optimization. So I just wanted to show you this as well, that is also available. And on November the 20th, I will talk again, and I will introduce this in a little bit more detail. And we also have developed another instrument which has the same technology integrated like the Tycho, which is called Prometheus Panther. So it has label-free nano DSF, but it also carries a new type of DLS that we have developed in Munich that is currently the fastest on the market, and it allows some new ways to analyze your sample. I will tell you more about this on the 20th. From 1 nanomolar to 1,000, 1 nanometer to 1,000 nanometer, we can do very precise sizing. And we also have back reflection in there that can be used to measure large aggregates. And the advantage of this is with the label-free nano-DSF and these DLS and back reflection optics, we can completely separate aggregation from unfolding. So we get very clean data regardless of aggregation, even for multi-domains or multiple proteins in solution. So it's, I think, very interesting. And yeah, I would love to tell you more about it on the 20th. So we can do stability characterization, conformation, ligand binding, like here, similarity in QC. So it's also used for biosimilar profiling, buffer screening, formulation. So antibody formulations, for example, it's very commonly done with this instrument, or finding the right crystallization conditions. If you have a detergent screen, for example, you can do sizing, polar dispersity, KDB22 is for formulation as well. You can also do isothermal measurements over a longer period of time. Okay, now let's focus back on screening with Dianthus. So I want to present you a small project where we screened a fragment library against a protein that's called G9A, and that is a methyltransferase. So it's a protein that's depicted here that is methylating DNA. It works in the field of epigenetics and changes basically methylation and acetylation of a DNA. And yeah, we used it in collaboration with the Imperial College in London for a fragment screening. If you're interested in this assay, what I show you now in much more detail, then go to our homepage. We have an app note of this collaboration. Okay, so let's go forward. Here's the positive control of this screening assay. You see here on the left side, these two pictures, that is a single dose hit of SAM, so a positive control. You see a very nice separation. You see smooth curves, it means no aggregation. You see reproducibility, so very nice data quality. Then we measured also the KD, was the concentration range of the ligand, and we measured 13 micromolar affinity. And I want to show you one of the fragment hits as well here in more detail. We see here as well, very nice data quality. There's a little bit more separation, but that doesn't really matter. And when you look here at the affinity experiment, we don't get complete saturation. So we measured all these fragments with 1 millimolar highest concentration in the assay. So we loaded the entire plate with 1 millimolar of all fragments, and that's what we got. And we see a very nice separation, but it's not saturated. So we concluded the affinity of this fragment here is equal or taller, more likely taller than 2 millimolar. So it's an extreme weak binder, but we can very reliably find these. And we can also very reliably exclude false positives as well. Yeah, that is basically the raw results data of the entire screen. And just to show you what you're seeing here, you remember our single dose hit always has a negative control and a noise level here in pink. And the pink bar is behind all these non-binders. So it's basically all negative. Everything which is outside of this bar has potential or is positive. So everything which is outside gets automatically classified in different categories. That can be, okay, in green, we have aggregation. Normally we go forward by spinning them down or changing the buffer. And if you don't have aggregation, then it's a very potential hit. So these are actually very nice. In blue, you have hits. We have clean data, reproducible, and a sufficient amplitude to conclude on binding. In yellow is a very solid positive control, which you see here. So binders, very promising ones. This one here, these two here, yeah, whatever you see here as well. It's the green ones I show you here, our classification. So you get some kind of this kind of chart. In gray are all non-binders in this assay. In blue, we had hits. Yeah, we had 89 hits in the screening. We screened in total 2,500 ligands in duplicate. Then you have here a few with aggregation. There are some with autofluorescence, and then some that are not reproducible or where one of these trick traces shows aggregation, the other not, something like this. So that means you have clean hits here. As next, maybe work on the aggregation, and then you can look into how much potential these potential hits have. You exclude all the rest. Well, that is the result of the full screening, that you have the numbers of the screening. 5,780 data points were acquired within 8 hours of instrument time. So you could fully automate this without a problem. That runs in half a day, of course, with pipetting. Or maybe you interlace this, then it's really half a day. 20 microgram of the target protein were used for both the single dose screening and also the affinity screening of the hits afterwards. So you really don't need much of your target protein, which can significantly reduce maybe the costs that you put into such screenings. Yeah, the 88 hits were identified ranging from 100 to 350 Dalton, these fragments. And yeah, I don't want to tell more details. I think that is the core message. Here are all the points again. I thank you very much for listening to me, and I would be very happy to get your questions. And in case you have further questions after this talk, please note down the email address of my colleague. Thank you very much.
Yeah, thank you very much, Rene. And may I request Deepak to provide this felicitation certificate. And Rene, what we will do, we will go directly to panel discussion along with Sampada, who is available with us. So here is a small token of appreciation in the form of certificate. Thanks for your excellent talk, Rene.
Great, thank you so much.
Okay, so Deepak, we can take Sampada online, and I hope she is there, and we can go for questions to Sampada as well as to Rene.
Yeah, so Sampada, may I request you to switch on your video so you can be also in the same frame? Yeah.
So here is a first question, Sampada, for you. And again, thanks for your talk. Sorry for small glitches to all our audience, but I think she could able to pull it once we went off from video. So very good presentation, Sampada. It has generated a lot of interest. So a question for you is, biotherapeutics tend to have a lot of impact of heat and stress during transport, storage, or its uses. So whether Shimazu aggregator can help in studying the impact on particle distribution or aggregation during these stress conditions? Can you please elaborate a little bit?
Yes. So Shimazu aggregate sizer can find out, or it can help you to find out the ways in which you can study the stress conditions, that is the temperature and the stress stimulation conditions, by which you can set up your protocol and you can set the storage conditions accordingly. So this is how by which we can study the size distribution for this aggregate analysis and their impact on a heat at various temperature studies by using the different accessories which I have shown mine in the presentation.
So, so just a follow-up, very quick question. So did we generate any data at Shimadzu for any of the biotherapeutics, especially like immunoglobulins or anything for this?
Yes, the data shared with you in the presentation was generated.
Okay, all right.
Was generated on the Shimadzu instrument by aggregate sizer only.
Okay, great. Thank you.
In Mumbai also we have generated few data.
And any precaution, Sampada, do you suggest there based on the troubleshooting required and based on your experience?
As the troubleshooting, because this basically the instrument is very simple to handle, similar to data for other particle size analyzer techniques or other complicated techniques. So as compared to that, this is very simple. There's a similar to we handle the UV visible spectrophotometer. Similarly, we can handle the microcells or the bat cell over here. So troubleshooting part is very less in case of the aggregate sizer.
And just the last question here on this particular topic. So the range you have mentioned from the FDA is 0.1 to 10 micrometer?
For the instrument range is 0.2 to 10 micrometer.
0.2 to 10, and it works well in that particular range?
Yes, sir.
Okay, okay. Great. So thanks, Sampada. And Rene, a question for you is like you have mentioned mainly on the ligand binding and target base. So that is like describing biochemical assay. So just a very basic and new question. Can the same thing you be used like in non-labeled or unlabeled manner for cell-based assay also?
Now you can hear me. So cell-based assay and let's say truly label-free. Yeah. In high throughput, so in the dianthus, we need the dye. Yeah. So then it's not 100% label-free. But we have a low throughput instrument that can do like 10 kDs per day. Yeah. That is truly label-free. So it doesn't need a dye. It measures intrinsic fluorescence of your protein. And if your dye, for example, if your ligand is not aromatic and exhibits not out of fluorescence in the same range that we measure the intrinsic fluorescence, you can measure entirely label-free in solution. So that is possible. And the question on if cells are possible, it depends on the type and the size of cell. So we look at, let's say, we look at very small volumes, and we need a certain number of targets that we can see in this volume. If the cell is too high, then the noise will, if the cell is too big, the noise will increase. So for some cells, it works well, where we expressed receptors on the surface, and we have ligands that bind to these receptors. We have some app notes on this. And for some cells, it's just too big. Like E. coli, for example, I would not recommend to try this.
Thank you. So should we consider that it is more focused towards biochemical assay?
Yeah, it's made for biochemical assays. Yeah, for purified samples in solution, yes.
Okay.
Or
And
1 second. Yeah, one more thing. You can also measure in lysates. So in case you cannot purify your protein, you can prepare lysate, you can measure in there. Yeah.
okay, another question is, though your specific plate 384, I understand they are meant for to avoid non-specific binding. But what
Yeah.
about other things which are used, which are not like, let's say, supplied, keeping in consideration non-specific binding, let's say tips, pipettes, what is like essentially will be used, are microcentrifuge appendoffs where things are taken. So how to avoid those kind of challenges, non-specific binding, to overcome any false positive or negative?
Okay, so let me repeat the question that I understand it 100%. So is there a way to understand if my sample maybe gets stuck to my preparation tube or pipette and it doesn't even reach the plate? Yeah, then we have concentration gradients, we have bias data, these kind of things. Is there a way to understand this? Yeah, so yes, definitely. For this, I would like to draw a figure actually, but let's say we have a ligand concentration 1-to-1, let's say 16 or 8 data points, and we have the target with the same concentration. That means we see the target with the same fluorescence intensity in all data points. If now the target is sticking to our labware pipetting solutions, not as much target will reach the plate. So the higher we go with the ligand concentration, and if it binds, then we passivate basically the target from our equipment, and we get more target in the final plate. And when we look at our raw data, we see gradients, fluorescence gradients, and then they can tell us precisely, okay, something is getting lost on the way, or we have ligand-induced quenching, for example. It's also what we can measure. So yeah, there are different ways to understand this on the way.
Okay,
yeah.
Thanks. So question for Sampada is like, what way it is possible or how it is different when for the large molecule it is in finished product where the buffers and other things are there in the intended formulation, versus when protein is in its native nature? Sampada, if you can highlight any input on that.
Sampada, you are muted. I mean, your line is muted. Can you just unmute and
So, Sampada, you got the question, or you want me to repeat?
Sir, can you please repeat?
Yeah. So the query is, when, let's say, protein, say for example biosimilar, if it is in the finished product in the formulation form, let's say whether it's a suspension or in the solution injection form, versus when it was in native form, when it was expressed in the pure form, when the formulation is not prepared. So can it differentiate aggregation in both the form, and how it is done, like using this Shimazu instrument?
Okay. It cannot differentiate the aggregation between the different forms, whether it is in biosimilar form, in the second form, because for the instrument, it is like a size, a particle it is considering. So it cannot differentiate. It can measure only the size distribution based on the aggregates or whatever the particle it is observed.
And what are other orthogonal techniques which are preferred, in comparison to this particular technique?
Like RMM, that is resonance mass measurement, is also done with this. Sometimes the light obscuration also, but depending upon the range of analysis which you want to test.
Okay, okay. Thank you, Sampada. And then I have one more question for you, which is very, very basic in nature. So when we are mentioning binding and KD values, so whether particularly this technique is complementing or no way it is associated with the SPR, what it gives binding with the target?
Okay, I'm not sure if I got the question right. So how it complements, for example, SPR?
Yeah, how it complements for the surface plasma resonance. Yeah.
Yes, yes. Yeah, so first of all, of course, SPR is a fine technology, but we sell a different technology. So in SPR, you immobilize your target. If you have, for example, a challenging protein, let's say a transmembrane receptor, they are very hard to immobilize. So you go down in pH, you unfold them, you immobilize, and maybe they're unfolded on the chip. Something like this, some proteins don't tolerate this. You can address these types of molecules in our solution because you don't immobilize. You can make them happy and measure them and keep them in this solution. Another thing is mass independent. So Dianthus is mass independent. That means especially for small mass changes, if you have maybe a GPCR or let's say a transporter for ions, and an ion binds in the ion channel, there's no mass change basically. You have just an ion in there. So SPR cannot see it, but you might have a conformational change, and that changes the reporter fluorescence that we are measuring. So we can address these kind of targets that show like a very small mass change. So that's another thing where Dianthus can complement SPR. And the third thing is SPR measures very reliably, also in the presence of, for example, irrigation. Maybe it gives more optimistic false positives sometimes. So you can do a secondary screen after SPR, for example, with the Dianthus to narrow down the really potential hits to complement and to filter this a little bit more. So in this way, you can complement for challenging targets, for small mass changes, and to clean up your data a bit. Yeah.
So thanks. And another question for you from Peter Annert. So welcome, Peter Annert. Nice to hear a question from you. So that is like, can binding experiments be performed with membrane vesicles expressing certain transporter, for instance?
Yes, that's possible. We have some app notes on this as well that describes this. Maybe visit our homepage or write us an email. I mean, that might be better, then we can send you what you need. Yes, snare proteins, for some interaction of snare proteins was analyzed. Then we have some receptors that we expressed integrated on the surface of a vesicle, for example. Then something else can bind to this, or we have a receptor formation on these vesicles. That's all possible and doable. Of course, these are more, these assays need a little bit more optimization and trying and good controls to get through than just a protein small molecule interaction. But they're definitely possible, and there are very nice published examples for this.
Thank you. And how fast it can be established as a new technology in the lab? A general question.
I can train you in 1 day, so you can use it, no problem.
Okay, thank you.
Yeah.
So that is operation part, but then we have to, like, as you said, we have to understand the basic part, buffer uses and other things. But operation wise, you are telling that it is within a day it can be established.
Yes, there's almost
Okay.
no optimizations out of the box.
Okay.
It's rather simple. Yeah.
Thank you. So I do not see any more question. Deepak or other fellow panelists, if you are seeing any other question, either for Sampada or for Rene, please bring it. Or if you yourself have any query, otherwise, Deepak, over to you.
I see one question from Shrikant. He says, in most of the kit-based assays, the data is not reproducible with the protocol suggested by vendor companies. So what can be the practical challenges, if any?
Is it for me, the question?
Looking like for
Yes.
Yeah.
Okay.
Yes.
Yeah, reproducibility. So there are very nice science papers on the reproducibility crisis in science, that it's very hard to reproduce what somebody else is publishing. There are numbers in all kinds of fields like chemistry, biology, and all kinds. And there were questions, how likely is it that I cannot reproduce my own essay or somebody else? And the numbers were up to 60, 70%, was extremely high. So to avoid this, I recommend the small blue instrument that we sell, the Tycho, to analyze in detail the quality and batch similarity of the samples that you're working with. The more similar these are, the higher reproducible will be what you do. I hope that answers the question.
Yeah, very much. Thank you. So hope, Shrikant, that answers your question. So finally, JJ, any questions from your side?
Yeah, I had a quick question for Sampath.
Yeah.
How does your device compare to a Mal's device, and can it be coupled to chromatography?
Sir, actually, it cannot be compared with the Mal's technique because the measurement range for the Mal's is different than that of the aggregate analysis, aggregate sizer. So generally, the Mal's analysis are performed for the dimers and the tetramers, which is in the range of nanometer. That is less than, if I say, it will be about 50 nanometer, less than 50 nanometer, whereas aggregate sizer works about 100 nanometer range.
Okay, thank you.
All right, so if there are no further questions, then would like to take this opportunity to thank all of our esteemed speakers, starting from Sampada, Sanjeev, and Rene. Thanks a lot for a very wonderful session we had. I mean, great talks, very informative, and a very interactive panel discussion. So thank you, all of you. And as I said earlier, we would really like to have you all in person next year when we do a in-person meeting, hopefully with COVID-free era. So once again, thank you to all of our speakers. And finally, let me invite the moderator, Dr. Ravi, for his final concluding remarks. Over to you, Ravi. Thank you.
Yeah, thank you, Deepak. And thanks, Professor Sanjeeva, Rene, and Sampada. I know it's not only weekend, but here in India, being festival time, it really shows like a lot of interest and enthusiasm where people came out and from their during festival type busy schedule and attending this session. So I thoroughly enjoyed all the 3 sessions, and certainly, as Deepak rightly mentioned, hope next year will be a normal year where we can meet in person and we can like have these discussion in person. And lastly, we will be posting if there is any other follow-up question and audience request. Please, I think everybody has shared their email IDs. Feel free to reach out directly or through common email ID for further question. So that's it, Deepak, from my side.
Thank you, Ravi, for for moderating the session so well, and would like to thank all the speakers once again, all the panelists and the attendees. And now would request all of you to. I'm just going to launch the on your screen for all the attendees. You can see one webinar poll. I mean, just with few questions. Please, you can. Submit your responses and it will be on for a couple of minutes. So once you submit your responses, then we will quickly have 3 to 4 minutes we'll spend on viewing a video, both from Shimadzu and then first from Nanotemper and then from Shimadzu. And then we will go for the last and final activity of our webinar, which is we'll go with the draw. So on your screen, you will see a few questions. So please submit your responses. So on your screen now, it's called Webinar 14 poll. So you can just submit your responses, a few very basic questions about the webinar, and then we are ready to go for the next session. Yeah. And I always receive this request from panelists, so even they are allowed to participate. So feel free to submit your responses. And while everyone is just participating in the webinar poll, let me just quickly mention that next Saturday, again, we have got a very interesting webinar lined up from Biotherapeutics Development Team, that is from CMC. And I think we have very interesting talks lined up with a talk from Satish from Moderna Therapeutics and a couple of other interesting talks. So would like to request all of you to block your calendar for next week, next Saturday, again, same time from 8:30 to 11:30. And looking forward to all of you joining in the next webinar again. So another 30 seconds, I will just keep the poll on, and then I'll end this poll. All right, so we'll officially end the poll. So I'm just going to press the end button here, and it will take a few seconds to download the results, and we should have the results with us. All right, so I have the results with me. So thank you for all of you for participating in the webinar poll. Now let's quickly go through this two small promotional videos. First from Nanotemper, and then from Shimadzu, and then we will go ahead with the webinar poll draw. So Ravi, you can just confirm if you're able to hear and see the video. Yeah.
Yet to appear. So Deepak, video is yet to appear. Can you hear, Deepak? Okay, seems some technical glitch. Deepak?
Yeah, I don't see his microphone being on
Yeah,
anymore.
yeah. Maybe we will wait for a few seconds. I think he should be back soon.
Yeah, I think he's logged out.
Mhm.
I think it's not able to launch it. So just allow me a few seconds. Be there, Ravi. I will immediately start that again. Yeah, just
Not
give
a
me a few
problem.
more seconds. Yeah, thank you.
Not a problem. Deepak, if this first video is not working, we can change the sequence if you can hear me.
Yeah, thanks, Ravi, for your patience. Just allow me a minute more. I think I'm just trying to fix it. Just I think 30 more seconds, and we should be good.
No problem. No problem.
Thank you. All right, so yeah, hope Ravi, you're able to hear me.
Yeah, but there was some.
Yeah, is it better now? Okay.
Yeah.
So for some reason unknown, I got logged out. So and it was not allowing me to join again. So let me just try now. And there we go. Just confirm if you are able to hear me. You're able to see my screen?
Yes.
Okay.
Yes.
Great.
As the product manager of the Prometheus, one of the highlights of my job is to hear from customers about how the Prometheus and its nano-DSF measurement are making a difference in their development and characterization of biologics and biotherapeutics. However, because of the complexity of biologics and biotherapeutics, We know that more than just a DSF measurement is necessary to be able to characterize and understand these molecules. And we have been constantly hearing from customers about how they wish that they could apply the same level of quality of the Prometheus and the nano-DSF measurement to these other biophysical approaches. And this is where we have gone back and hit the drawing board. When thinking about what set the nano-DSF technology in Prometheus apart from other DSF measurement approaches, and what we hoped to achieve when thinking about integrating and implementing new biophysical measurement techniques, what we kept hearing over and over again from our customers was how the accuracy, sensitivity, limit of detection, precision, resolution, and these other properties of the nano-DSF measurement are what make it the best-in-class approach and the gold standard for characterizing the DSF stability of their biologics and proteins. And it is precisely those parameters and properties that needed to be maintained, not only for the nano-DSF approach, but also any other biophysical approach that groups are using for their biologic and biotherapeutic characterization. And that is precisely what we worked on when integrating our DLS optics. When talking with our partners who have been testing the product in the field, some of the biggest rave reviews and most important feedback that we have received from them are how the simultaneous measurement and acquisition of the nano-DSF alongside DLS during a single thermal ramp helps them to understand their molecules, their biologics, and their proteins in greater levels of depth and detail than were previously possible. And without any further ado, I would like to introduce you to the Prometheus Phantom.
All right, so thank you. I would like to thank Nanotemper for providing us with this promotional video. Let me quickly, in the interest of time, let me quickly check for the Shimadzu video as well. Okay, so Ravi, just confirm me once you're able to see. Yeah.
Sure.
The biopharmaceutical market is growing rapidly due to an increase of novel therapeutics and biosimilar drugs. Contributing to this growth is the development of therapeutic monoclonal antibodies to treat a variety of serious diseases. Because biopharmaceuticals are made in living mammalian cells, the increased interest in biologics has resulted in significant growth in the cell cultures market as well. In particular, Chinese hamster ovary or CHO cells are the most common mammalian cell line used in the production of therapeutic proteins. Ensuring that the culture media mix is optimal for cell growth is vital to drug development and the quality of the final product. Analysts must frequently test to make sure the cell culture media contains the precise balance of glucose, glutamine, nucleic acid, vitamins, and other biologically important compounds and primary metabolites. Developing monoclonal antibodies is especially difficult because scientists are working with extremely complex molecules that have many attributes, all of which must be analyzed. These complex molecules can produce thousands of combinations of modifications. This means analysts must conduct numerous detailed analyses throughout the drug development process. Researchers need a fast, efficient, and holistic way to test cell culture media during process development and bioproduction, in both traditional batch mode and in continuous manufacturing. However, conventional electrode and sensor-based methods measure only certain analytes and allow a limited range of parameters to be monitored. A better solution is using higher resolution technology, like liquid chromatography combined with mass spectrometry. LC-MS is more accurate, has greater sensitivity, and allows the simultaneous measurement of many cell culture components and their metabolites. These combined capabilities are not possible with current conventional techniques. For the most effective cell culture analysis, scientists need a total solution that provides fully optimized chromatography conditions to allow separation of maximum number of compounds in a single run, fully optimized and validated MRM conditions for each of the components to ensure specificity and sensitivity, an LC-MS platform that allows fast scanning and polarity switching to detect components in both positive and negative modes, and data reporting that allows easy visualization of cell culture media component trends. Shimadzu's triple quadrupole mass spectrometer LC-MS 8060 and cell culture profiling method package make analysis of cell culture media fast, easy, and thorough. The LC-MS 8060 provides high throughput that is fully integrated into analytical workflows. The cell culture profiling method package enables better monitoring of the fermentation process, especially in continuous manufacturing where the media need to be constantly measured and rebalanced. This is important because today's biopharmaceutical developers need to grow cells over a longer period of time in a continuous manner. And scientists using the cell culture profiling method package can analyze up to 95 components simultaneously. Biosimilar manufacturers also benefit from using the cell culture profiling method because it makes it easier to ensure their media mix is similar to the original drug maker's media. Additionally, cell culture media manufacturers can also utilize the method package for optimization of ingredient amounts and reduce media lot-to-lot variability using better analytics by LC-MS. And stem cell researchers can use the cell culture profiling method to optimize specialized media needed to grow and maintain viable stem cells for stem cell therapy. The LC-MS 8060 with the dedicated cell culture profiling method package is the solution for biopharmaceutical analysis that researchers have been waiting for. For more information on how Shimadzu can help you improve the efficiency of your biopharma analytical workflows, visit www.labtotalbio.com.
All right, so that was a promotional video from Shimadzu. So we'd like to thank team of Shimadzu also for helping us with that.